We're going to use the following video to help you further understand membrane transport as it occurs within the cells in our body. So, of course, we began by discussing the fact that the plasma membrane tends to be the barrier through which we need to transport most substances, especially when we're talking about the transport of substances between our cells and our extracellular fluid. And we, of course, recognize that it is selectively permeable, so some things are able to pass through unassisted. Now, based on this concept, we find that those things that can pass through unassisted actually undergo what's called non-carrier mediated or simple transport. They don't need one of those membrane proteins in order to pass back and forth. However, as chemistry changes from some of these solutes and solvents, we find that they may require carrier-mediated transport, also referred to as a facilitated transport process. In addition to this, there are some substances that need the use of ATP to assist with their movement, and this would be referred to as an active transport process. But the majority of substances we'll study are those that don't require the hydrolysis of ATP in order to actually go through transport, and those are all called passive processes. We started off here with filtration. Filtration is the type of process by which you have a series of forces here within the inside of the capillary and the ICF and the ECF around the capillary. And what those result in is the ability of both fluid and small solutes to be pushed through the membrane of the cells that form the capillary wall and into the ECF. And this is a variety of solutes that we see that we can actually pass through the membrane in this method. The kidneys are the best example of where you'll see filtration occurring. And with the kidneys, that of course is taking fluids and solutes from the blood, pushing them through the wall of these capillaries and actually into a space where they'll be converted into the early stages of urine. We next move to simple diffusion. So diffusion itself is just the movement of particles from a place of high concentration to a place of lower concentration. What makes this simple is that the chemistry of these particles do not require any kind of a membrane protein for their transport. Since these particles are moving from high to low concentration, this is actually a passive process. You don't require any ATP to facilitate this motion. The idea here is that solute particles will just continue to move down this concentration gradient until they reach equilibrium. As you can see right over here, there are a number of factors that influence how diffusion can work across a membrane. As of course you can see, when temperature increases, all particles move faster, so they just diffuse more quickly. We also find that those who have a greater molecular weight tend to diffuse at a slower rate than those who have a lighter or a lesser molecular weight. We also find that the more steep the concentration gradient, meaning the greater the difference between the high and the low concentration, this can result in a faster diffusion. And we find that increased membrane surface area increases diffusion rate. The chemistry here of these individual particles and solutes is what allows them to actually go through simple diffusion as opposed to facilitated diffusion. Small molecules that are nonpolar and hydrophobic are the ones that are best able to interact with the fatty acid tails of your plasma membrane, thus are unimpeded as they begin to pass through. And this, of course, includes anything that is lipid soluble. So this is why certain hormones that are cholesterol or steroid based can pass directly through the membrane without utilizing any kind of a protein. The example that we use right here in this image is oxygen. Oxygen gas is nonpolar. Right? It's also very small. And because of its chemistry, it doesn't get stopped by the fatty acid nonpolar region of your plasma membrane. The other thing that we see right over here is there's a difference in concentration on either side of the membrane. Here in the extracellular fluid, you see there's a higher concentration of oxygen than exists here within the intracellular fluid. This is what's going to initiate the ability of oxygen to diffuse, to go from a high to a low concentration. And so in this example, you see the direction of its movement will always go from the ECF, where it has a higher concentration, to the ICF, where it has a lower concentration. So you can just see how individual molecules will begin to pass from ECF to ICF. Now, of course, as we said, this is going to occur until these particles reach equilibrium. But we're not actually going to see that occur here in the cell because as the oxygen enters the cell, it's going to be used up and converted to CO2. That way you continue to maintain this gradient and oxygen just continues to be able to simply diffuse into the cell moment by moment. Again, there's no ATP required for this process. It is passive, and that is because, again, it's the concentration gradient that is driving the direction of movement. Another kind of passive process that we see is osmosis. So osmosis is essentially still diffusion. It's still the movement of something from high to low concentration, but it's specifically the movement of water, not just any solute. So of course, we're still looking at a selectively permeable membrane here. The example would be our cells. And what you can see in this example over here is that we've basically got a tub, right, or a beaker set up. This dotted line represents a selectively permeable membrane. And what we're following primarily is the movement of water back and forth. Right? So the idea here is that even though we say that water needs to move from a high to a low concentration of itself, it does that by being attracted by solutes within a certain environment. And so we see that water moves from an area of lower solute concentration to an area of higher solute concentration. So here on side B, you see that there are smaller and fewer solutes in this particular region. Over on this side, we find that there are more and larger solutes. Now, as a result, the greater number and the larger molecular attractive force of these solutes that you see tend to pull the water from side B over to side A. And as that begins to happen, we find that it actually causes a lot of those solutes to spread apart, giving them more space between one another. But at the same time, it actually begins to increase the water level on side A. So the idea, once again, is that you do reach a state of equilibrium here, but now it's equilibrium between the solvent and the solute on side B, and equilibrium of the solvent and the solute on side A, not necessarily equilibrium between the two sides, right? Now the idea here is that you do have two forces associated with osmosis. You have osmotic pressure and hydrostatic pressure. Osmotic pressure is going to be the force that actually pulls the water across, and hydrostatic pressure would be something like air pressure or gravity that's actually pushing down on the water, trying to prevent it from rising any higher. So the idea here is, in regard to osmosis in our cells, our body cells are going to be altered by the osmolality or the concentration of our body fluids. So osmolarity or osmolality is the amount of dissolved solutes per liter of solution. And of course, we often reference this as tonicity, right? The ability of a solution to affect the fluid volume and the pressure within a cell. The different types of solutions that we describe for our body are either referred to as isotonic, hypertonic, or hypotonic. And you have to remember here that when we describe a solution like this, we are always referencing extracellular fluid, right? Again, and we are always comparing the osmolarity or the tonicity of the extracellular fluid to the cell's intracellular fluid. So let's take a look real quick at how each of these solutions could affect a cell. So what I have over here are 3 cells. Right? And for each one of them, I'm showing you that there is water both inside a cell in the ICF and water outside a cell in the ECF. And initially with each one of these, I'm showing you an equal amount of water, so 3 water molecules, on both the ICF and the ECF side. The other thing I'm showing you here is that the tonicity on the inside of each cell is about 0.9% sodium chloride. Now, of course, this is only referencing sodium chloride, but there are a number of solutes within the cell. So, of course, what this is showing you is that while this is 0.9% sodium chloride inside, you would still see about 99.1% water essentially making up the balance in just this really simple cellular example. Now again, the plasma membrane is a selectively permeable membrane, and water is able to pass back and forth across this membrane in small amounts unaided, but in large amounts you'll often utilize an aquaporin, which is just a membrane channel that allows water to go back and forth pretty quickly in larger volumes. So let's take a look right here at what happens when you place a cell of 0.9% sodium chloride ICF into a solution of your body, which is 0.9% sodium chloride. Now, the idea here is that these two osmolarities are actually identical, both between the ECF and the ICF. And so you'll find that neither one of them exerts a greater osmotic force on the water that exists on either side of the membrane. As a result, you'll have a pretty equal or net flow of water back and forth across the membrane. Now, this kind of a solution here around the cell would be referred to as an isotonic solution. So iso meaning the same effect or pull on water, which is both within the ICF and the ECF. This is the best environment for your cells to be in. If you ever look at a saline drip pouch at a hospital, you'll find that it oftentimes will read 0.9% NaCl because that's what you want flowing through your body. You want to make sure the balance of water in your blood plasma is pretty similar to the balance of water within your cells. Now, for another example, let's assume that we take your cells and we now place them in a full 1% sodium chloride solution. Well, what we refer to this as is called a hypertonic solution, and that's because it has a greater osmolarity here in this solution than it does within your cells. And because this is an area of higher solute concentration, it actually attracts the water from inside the ICF and pulls it out into the ECF. Now, of course, the greater the concentration of sodium chloride gets out here in the ECF, the even greater pull it's going to have on water. We look at the opposite situation over here, where if we place these 0.9% NaCl cells within a solution that is much lower in tonicity, so 0.1% NaCl, then that has the opposite effect on water. The higher concentration of sodium chloride still exists within your cell, and so water from the ECF gets pulled into the intracellular fluid. This is what we refer to as a hypotonic solution. Well, let's start with this example first. In this example, if water is being drawn into the cell, then what we find is that that's actually going to cause a cell to start to enlarge. And you can compare this erythrocyte, or red blood cell, to this normal erythrocyte, which would be found in an isotonic solution. This one's a little bit more flat, and this one is starting to swell up. Now, these can actually swell to the extent that they actually lyse or burst, as we would say, which eliminates the cell itself. Now, the other situation was the hypertonic solution, and with the hypertonic solution, you remember that water was drawn out of the cell, and as a result, you can see it starting to shrivel and wrinkle when compared here to the isotonic cell, or the cell in the isotonic solution. And we often refer to this as crenation, or the cell will crenate as it begins to shrivel. And so again, this all reflects the ability of water to pass back and forth between the wall here of the cell, so that cell membrane. And of course, the ability of water to do that is reflected based on the relationship of a solute concentration both outside the cell and inside the cell. And one more time to emphasize, the terms hypotonic, isotonic, and hypertonic always refer to the solution around the cell, so the ECF. So we finish here looking at carrier-mediated transport in this video. So again, these are going to be solutes that are transported across the membrane but have to utilize some kind of a protein carrier or channel in order to do that. Now keep in mind that all these carriers and channels are specific, and so only certain chemicals or ligands can pass through each type. And of course, the speed of this is going to depend on how many transport proteins are available. And so, of course, if you only have 5 transport proteins for a particular substance that are full, then as you begin to pass substances through one solute protein, you'll see the concentration begin to rise as it passes through the membrane. But then as it's filling up 2, and then 3, and then 4, and then all 5 of those available transport proteins, then you reach a transport maximum where you can no longer increase the speed of transport because all proteins are now being used. So there's a few terms we use to classify the proteins and the type of transport that they can facilitate. First off, we look at the direction of transport that's taking place within each protein. A uniport carrier is one that only transports one type of solute in one direction. A co-transport carrier is one that's transporting 2 or more solutes simultaneously in the same direction. We tend to call this a symport protein, again, sim for same or like. And the third is counter-transport. So it's when you have a carrier transporting 2 or more solutes simultaneously, but they go in opposite directions. So this is the opposite of symport. We refer to it as antiport. We're also going to say here that carrier-mediated transport can be referred to as one of 3 types of mechanisms. So not just directions here, but mechanisms themselves. So first off is facilitated diffusion. So again, this is diffusion, which means a solute is still being transported from a high to a low concentration gradient. So again, this is down its concentration gradient. And as long as it's diffusion that's taking place, it's still going to be passive, no ATP required. Primary active transport, on the other hand, is going to require ATP. And this is because the solute that's being transported is done so against its concentration gradient. So now you're actually taking it from a low concentration and pushing it towards a high. And that's the reason it needs ATP. A sodium-potassium pump is an excellent example of this. And then we're going to take a look at secondary active transport. And this is when you allow a solute to move down its gradient, but this is enabled because you are indirectly utilizing ATP hydrolysis somewhere else. And we're going to look at the example of the sodium glucose transporters for this. So here's a cell. We're going to look at 3 different examples, starting first with what's happening with the green carrier, then the blue carrier, then the yellow carrier. But I will go ahead and I'll show you the distribution of ions here around the cell. When we look at sodium, keep in mind that sodium tends to be in a higher concentration outside of a cell than inside. So that's going to be true here in this scenario. Higher sodium concentration over here in this scenario as well. And then higher concentration in the ECF in this scenario as well. So let's begin by looking at what happens with sodium due to this concentration gradient. Now, first off, we saw oxygen moving in something similar, but oxygen was a small nonpolar molecule, so it was able to pass directly through the membrane without any kind of a channel. The ionized characteristic here of sodium, however, it prevents it from being able to pass easily through the fatty acid and nonpolar region of your plasma membrane. It's essentially going to be rejected. And so just as long as you have some kind of a channel or a carrier protein present, sodium can move down its concentration gradient into the cell, and it does so passively. Again, because there's no ATP required for things to move from high to low concentration. Now, just to make sure all the terminology we're using makes sense with this kind of an example, since this is just one substance moving through this channel, this is called uniport. It is considered facilitated diffusion because it's moving from high to low, but facilitated because it uses a channel, and it is an example of passive transport. Let's move to the second example down here, showing you active transport. This is going to be primary active transport. The idea here is that this blue carrier is actually able to transport substances in 2 directions. Right? So the idea here is that you're going to take sodium right here that moved into the cell from a number of processes, and you're going to prevent it from building up in too high of a concentration by pushing it back out of the cell. Now, as you can see, if sodium has a higher concentration in the ECF than it does in the ICF, that means we're pushing sodium against its concentration gradient. Now, this particular carrier does that simultaneously by moving potassium. Now, potassium, and this is kind of new, actually has a higher concentration in the intracellular fluid than it does in the extracellular. But in this process, we take potassium and we also push it against its concentration gradient into the cell to try to maintain this high concentration inside. So you can see potassium moving in while sodium moves out. This is going to be an example of your sodium-potassium pump. So the terminology we use right over here is it is counter transport because you're moving substances in opposite directions simultaneously, but it's also referred to as primary active transport. This kind of transport requires ATP because it is moving substances against their concentration gradient. The third type of transport we look at is secondary active transport. Now, what we're going to see right here is that secondary active transport, when we look at this example by itself, looks a lot like diffusion. So let's look at the mechanism that takes place. Again, we're relying on the fact that sodium has a higher concentration in the ECF than in the ICF, as does glucose, as abbreviated GLU here. And so the idea is that this particular protein channel or carrier can actually transport both sodium and glucose from the ECF into the ICF through this particular channel. Now, because these are moving down their concentration gradient, it looks a lot like facilitated diffusion. But we refer to it as secondary active transport, and here's the reason why. In the cell, you'll find that this particular mechanism relies on the ability of a sodium-potassium pump to be running at all times. And that's because as this sodium begins to move into the cell, it starts to reach equilibrium. Without the ability of the sodium-potassium pump to run over here, normally in close proximity to this sodium-glucose transporter, then this movement is going to stop, which means both sodium and then glucose will no longer be able to enter the cell. But as long as your sodium-potassium pump is forcing sodium against its concentration gradient outside the cell, that enables this nice steep gradient here by which the sodium-glucose transporter can operate. This is an example of co-transport, where you have two substances passing through a channel or carrier, and this time they're moving in the same direction. So this would be a symport protein, while this was an antiport protein. In your notes, I've given you these same examples, and I think what you should do is sit down and read each example and begin to explain to yourself why each one of these would be a particular type of transport, and of course, ask yourself why it's considered a passive or an active process. So here we're just looking at the transport of some solute, right, through some kind of a protein carrier. Here we're looking at the sodium-potassium pump. It gives you more specifics here of how that pump actually works. And then the sodium-glucose transporter. Please feel free to watch videos through APR that show you how both the sodium-glucose transporter and the sodium-potassium pump work prior to trying to answer these examples. We'll end here with vesicular transport. Vesicular transport itself utilizes those vesicles that we discussed that are membranous organelles within the cell. And there's a few different types of vesicular transport that you see. First is endocytosis, which we refer to as the ability of a cell to actually bring matter into a cell using vesicles. Phagocytosis is probably the best known example of this, where you actually have cell eating. And this is showing you here a macrophage that actually is engulfing old red blood cells as it eliminates them in the spleen. And it does so by just basically binding with them and then sucking them in using a vesicle. Penocytosis works the same way, only this is bringing in small bits of ECF with little bits of solute instead of really large substances like what you'd see in phagocytosis. Now, while we're talking about phagocytosis, let's look at the process here by which this actually works within a cell. So here's your cell membrane right here and your nucleus, and there's the particle the cell wants to phagocytose. So the idea is that as it begins to eat it, as you could say. What you have here is the formation of what looks like a pseudopod, where basically you begin to pinch in on the cell membrane, engulfing this particular particle. Now remember that the membrane itself, being a phospholipid bilayer, right, is just going to wrap all the way around this particular substance until it actually brings it all the way into the cell. Now, this little package by which this particle is engulfed retains the same chemistry of the plasma membrane, and so it's able to interact with other membranous organelles within the cell. Initially, it's called a phagosome, right, because this is just associated with phagocytosis. And as it begins to move through the cell, in many cases, it will fuse with a lysosome. Now, a lysosome is a membranous cellular organelle that has enzymes within it that have the ability to digest. And so as the two fuse, they form one unit, a phagolysosome, in which this particle begins to interact with the enzymes from the lysosome, thus breaking it down. Once this is done, a number of things can happen with this substance, but here it's showing you that phagolysosome, with its membrane characteristics, can simply just merge with the plasma membrane once again, and then cause you to expel the product. This is called exocytosis, and we're going to look at it here in just a minute. But that's how phagocytosis is going to work. This is how your cells basically test an environment. It's also how they eliminate pathogens if they happen to be an immune cell. Now, sometimes you have a really specific kind of endocytosis in which any kind of a particle that is going to be endocytosed into the cell must first be able to bind to very specific protein receptors on the surface of the cell. And so we call this receptor-mediated endocytosis. It's like these particles have to know the secret password in order to get into a cell. Not just anything can enter. And so oftentimes this is what we're going to do with hormones within the cell, or this is how an immune cell would actually bind to a specific bacterium and begin to engulf it. And so you can see that basically those particles just begin to bind to these protein receptors, which activates the formation here of a pit, which then is going to close off and essentially just form what looks like a phagosome here, but we would just call it an endocytotic vesicle. What we've found in the body is that sometimes bacteria or viruses can hijack this mechanism. This is actually how influenza virus infects your cells. It looks a lot like another particle within your cells that's healthy, so it can bind to these receptors and just be invited into this vesicle, which then it begins to replicate and infect. So it's utilizing receptor-mediated endocytosis, but it's hijacking it essentially. And then finally, we see exocytosis. So exocytosis itself is where you're actually releasing material from the inside of the cell to the outside of the cell. So it's just the reverse of endocytosis. Here's the secretory vesicle. Again, a secretory vesicle is one that would have usually been made by either the rough or the smooth ER, and then modified by the Golgi apparatus, so it can then be released. So again, this is how you're making mucus, hormones, enzymes, even breast milk. And so the vesicle right here, having the same chemistry as the plasma membrane, is able to bind to the membrane using a series of proteins. It'll merge and open up, and then will actually release its contents. Now, this vesicle actually becomes a part of the plasma membrane, and so we can say that it essentially is replacing membrane regions that were lost during endocytosis. And so this video right over here does a really nice job of showing you how all these membranes and these vesicles actually interact with one another. So we'll have this open, so you can watch the video with me. Here we go.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane.
So this, of course, is showing you exocytosis, particularly of a vesicle that's been produced by the cell. And as you see through the illustration here, the whole reason that's possible is because the vesicle has the same membrane chemistry as the plasma membrane itself.
This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
Right. And so that ends our discussion of membrane transport. One last thing to mention right here is that vesicular transport of each kind tends to be an active process. So you tend to need to utilize ATP in order to make sure that this all occurs. So in review, you have processes that are both facilitated by ATP or no ATP, in which case we refer to them as passive or active. And then you have many processes facilitated with proteins of some kind, and then those that don't require any kind of protein. And the best example we saw there was simple diffusion. So as you walk back through each of these examples, ask yourself the many questions that were associated with terminology. And email me if you have any questions.